Hi, Biology World. This is Paulus here, and the purpose of this video is to give those students who missed any of the 12-1 class notes an opportunity to get them copied down, as I might have gone too fast, or maybe they were absent one day. In any event, I'm making a video so you can pause it and you can fast forward through it. Remember, all class notes need to be in Cornell note style, and they need to be numbered. The questions are going to go on the left, and the answers are going to go on the right, and they need to be numbered. All right. Also, I'm going to be going through these a little bit faster than I normally do in class because you can always pause the video. If you don't want to listen to me talking, I will have some things interesting to say. Well, maybe not interesting, but they're going to help you learn. But if you want to mute me, that's totally fine. But do understand that if you see me point at myself or point at the computer, that's when I'm about to hop to the next slide. And of course, if you're not done writing the things that are on this slide, that would be when you would want to pause the video. Okay, all right, starting off, Frederick Griffith was a scientist, and he studied the disease of pneumonia. And what he was doing is he had 4 experiments. He had a bacteria that were called, beg your pardon. Yeah, disease-causing strain of bacteria that were referred to as smooth. So when you grow bacteria, you grow them on a nutrient plate. It's called an agar plate. And they just have kind of a smooth appearance. Like if you could touch this little circle that is all the bacteria, and usually a little tiny circle is millions of bacteria that has a smooth appearance. Those are the ones that cause the disease. The harmless strain of bacteria grew into colonies with rough edges. So it looked like they have kind of a rough texture. Okay. So again, these disease-causing bacteria cause pneumonia and death in mice. These harmless bacteria do not cause pneumonia and, of course, do not cause death in mice. Moving ahead. Okay, so we did 4 experiments. Experiment 1 and 2 were controls. He injected mice with the disease-causing strain. And, of course, they died. He expected them to die. This is a controlled experiment. Experiment number 2, he injected them with a harmless strain, and of course they lived because they did not develop pneumonia. Experiment 3 also was kind of an expected experiment, a controlled experiment. Griffith heated the disease-causing bacteria, killing it. So remember, proteins get denatured and their shape changes when you expose them to heat. So basically, he killed the bacteria and their disease-causing ability by heating it. He injected the heat-killed disease-causing bacteria into the mice. The mice did not develop pneumonia, and they lived. The 4th experiment is what got a little weird, and he's probably scratching his head going, what the heck just happened? He injected the heat-killed disease-causing bacteria into the harmless bacteria. He mixed them. He didn't inject them. He mixed them. And he took this mixture, injected it into the mice, and they died. So the harmless alone, they lived. The heat-killed disease-causing alone, they lived. But when you mix the 2 together, the mice died. Again, head scratcher. What's going on there? What he concluded is that the heat-killed bacteria, the heat-killed disease-causing bacteria, passed on their disease-causing ability to the harmless strain. Okay. Now that's, just to clarify, that's all he knew. He didn't know how the mice died. He didn't know what was being transferred from the heat-killed disease-causing into the harmless. He didn't know any of that. He just knew what was happening. Okay? Moving ahead. He called this process transformation. That's when the harmless strain transformed into the disease-causing strain when they were mixed. And again, that's all he knew. He just knew that transformation took place. He did not know how it took place. Excuse me. It's been a long day. Other scientists that came after Griffith helped explain what was the transforming factor, and they worked with bacteriophages, viruses that infect bacteria. But all Griffith knew is that transformation took place. He did not know that it was DNA that did the transformation. All right, that's what Avery figured out. So Avery wanted to know which molecule did the transformation. And again, there are a lot more details about what Avery and his experiments did. He used enzymes. I didn't want to bore you with those details. Of course, you'll want to take a look at those details when you're reading and doing your reading notes. But just simply know that Avery was interested in knowing which molecule was involved in transformation. And the two camps were, it's either proteins, because proteins are really, really complex, or it's DNA, because DNA is really, really complex. So it had to be one of them. Okay? All right, my bad. Moving ahead. Avery's conclusion, he concluded that DNA was the transforming factor. Again, there are a lot more details into how he figured that out. Please read those details, but you don't have to take them in the class notes. Okay, relationship between DNA and genes. Basically, DNA is a really long molecule, and all the segments within this long molecule are called genes. DNA stores and transmits the genetic information from one generation of an organism to the next. And that's why, assuming you know who your birth parents are, you have a combination of characteristics between your parents. Half of the DNA in your cells is DNA from your mom, and the other half of the DNA in your cells is the DNA from your dad. And DNA, and we'll learn about this at the beginning of the next semester, DNA codes for proteins. Genes code for proteins. So the proteins, which give you your characteristics, are a direct result of the DNA that you have in your cells. So the proteins that give you some of mom's characteristics come from her DNA. The proteins that give you some of dad's characteristics come from his DNA. Okay, getting ready to bump ahead. Okay, please use this as an opportunity to practice with your notes. I'll tell you where to go look. You might even know the answer. Please don't fast forward and go to the next notes. Use this as an opportunity to practice. Okay, Griffith concluded that blank bacteria passed on their disease-causing ability to the blank bacteria. And what did he call that process? Take a look at number 4 and 5. Sorry, I'm yawning. Number 4 and 5 to answer that question. Yeah, so he concluded that the heat-killed disease-causing bacteria passed on their disease-causing ability to the harmless bacteria. And of course, that is called transformation. Avery and his colleagues determined that what was the name of the molecule responsible for the transformation? Again, if you're not sure, you might want to take a look at number 7. Yeah, you bet. It was DNA. That was the transforming factor. Okay, Hershey and Chase. So Hershey and Chase studied viruses that infect bacteria, and those viruses are called bacteriophages or bacteriophage. And these are, I beg your pardon, I'm going to need to go make a cup of coffee in a bit. The virus attaches, what do? They inject their DNA. The virus injects its DNA into the bacteria. And then what they do is they basically take over the bacteria and they say, you're not doing what you normally do. You're making viruses and you're making lots of them. And after you make all these viruses, you're going to die. So that's what bacteriophages do. They infect bacteria By injecting their DNA into it, the DNA is used to make more viruses, and then the viruses make the cell explode, and then all these other viruses go out and attack other bacterial cells. And this is the fundamental reason why viruses are not considered living things. They need cells. Remember, bacteria is a cell. It's a single-celled organism. They need cells to do their reproduction for them. The definition of an organism, well, there are lots of definitions of an organism. One of the characteristics of an organism is that it can reproduce on its own. Even yeast, they use cloning, and some bacteria use cloning. They don't use sexual reproduction. They still can do it on their own. Viruses cannot reproduce on their own. They need a host cell to do it. Therefore, they are not considered living, and they are not considered organisms. Moving on. Okay, so how does the virus reproduce? The viral genes produce many new bacteriophages. Eventually, the bacteria is destroyed. So what happens is the viruses are building up and building up and building up, and the cell starts to expand. And it starts to expand, and it explodes. And then all these hundreds of viruses or thousands of viruses go on, and they infect other cells. And that's how a viral infection begins. So the next question that they were interested in knowing was whether or not it was protein, indicated by radioactive sulfur, or DNA, indicated by radioactive phosphorus, that was being injected into the bacterial cell. And they used radioactive isotopes. Remember, an isotope is an atom that has more or fewer neutrons than the natural atom. And they used radioactive ones because you can actually see these things with an X-ray. How are we doing on time? I've got a meeting at 3:30. We're good. And you can abbreviate S, capital S for sulfur, and capital P for phosphorus. Totally okay. Oh dear. All right, moving ahead. Okay, so what's this mean? And I'm sorry you guys can't see this down here. It's blocked by my image. So this is what a virus looks like. A bacteriophage looks like. There are lots of different shapes of viruses. And all these little circles represent the protein coat. Protein coats have sulfur in them. So this virus finds a bacterial cell. It sits on it. It injects something into the cell. This material that was injected into the cell does not show any radioactivity. Therefore, there's no radioactive sulfur inside the bacteria. Therefore, protein was not injected into the bacteria. Okay? So this material is not protein because it does not have the radioactive sulfur on it. Okay, moving ahead to number 15. What else? If phosphorus was found, so you can see all the little circles here on the DNA inside. DNA has phosphate groups, so it's got phosphorus. And if you find radioactive phosphorus inside the bacterial cell, you know what was injected into the bacterial cell is DNA. All right, moving ahead. Okay, Hershey and Chase studied bacteriophage. What is a bacteriophage? And if you can't remember, take a look at number 10. Right, a bacteriophage is a virus that infects bacteria. How do bacteriophages reproduce using host cells? If you can't remember, take a look at number 12. Yeah, so they basically use the cell's machinery, its organelles, the workers, if you will, inside the cell. They use those things instead of the bacteria doing what bacteria do, and there are a variety of things that bacteria do and bacterial cells do. Say, you're not doing what bacteria do, you're making viruses. And it uses all the cell's machinery and all the cell's energy to make viruses, and that's why viruses are not considered living things. They're not considered organisms, is because they need other cells to do the reproduction for them. And what happens is it creates all these viruses and the cell starts to expand, and finally the cell blows up because you can't fit any more viruses in there. Then all the viruses run out to other cells, land on them, inject their DNA, and produce more viruses, and that leads to a viral infection. Okay, if they found radioactive sulfur in the bacteria, what did it mean? If they found radioactive phosphorus in the bacteria, what did it mean? Take a look at number 14 and 15. Sulfur means protein. They did not find radioactive sulfur. Phosphorus means DNA. They did find radioactive phosphorus. So they now knew that the bacteriophages were injecting DNA into the bacterial cells. Let's finish these up. Structure of DNA, it's made of monomers called nucleotides. Remember, nucleotides have 3 parts: sugar, in this case, deoxyribose, phosphate group, nitrogen base. Remember, the sugar in RNA, sugar in RNA is ribose, sugar in DNA is deoxyribose. That is one of the differences between DNA and RNA. Sorry, I am so tired today. Okay, there are 4 nitrogen bases. You can use the abbreviations if you know that A means adenine, G means guanine, T equals thymine, C equals cytosine. Okay, time to move ahead. DNA backbone. So the DNA's backbone, remember it has 2. This is the sides of the ladder, is referred to as a sugar phosphate backbone because it's the sugar bonded to a phosphate, bonded to a sugar, bonded to a phosphate, bonded to a sugar, bonded to a phosphate that creates the sides or the backbone of the ladder. And it does it on both sides. Sugar phosphate backbone on both sides. Then it's the nitrogen bases that make the steps or the rungs, RUNGS, fancy word for step, of the ladder. All right, Erwin Chargaff. He was a scientist that figured out, and he tested a whole bunch of different types of DNA in different types of organisms, that every time he found out that the percentages of adenine, so let's say, I don't know, let's just make it nice and round. No, that wouldn't work. Let's say the percentage of adenine was 40%. Okay. Consequently, the percentage of thymine was, and it might have been like 39.8% or 40.1%, but they were pretty much equal. And then, of course, if we're talking 40 and 40, that would have meant that cytosine was about 10% and guanine was about 10%. So he says, well, if you have the same percentages, that must mean that every time you have an adenine, it's bonded to a thymine. Every time you have a cytosine, it's bonded to a guanine. So it's a 1-to-1 relationship, equal percentages. And of course, this information helped Watson, Crick, and Wilkins figure out that the bases are what form the rungs, and that they aren't pointing outwards, that they're actually pointing inwards in their model. Moving ahead. Okay, Rosalind Franklin. Hope you guys enjoyed the movie. Remember, there is an hour-long version. It's called Photo 51 that has a lot more interviews, talk a little bit of trash about Watson because he deserves to have trash talked about him. Did not give her the credit that she was due. She used a process called X-ray diffraction. That's the famous Photo 51 that you can see there. She figured out information about the structure of DNA. She did not get credit for the structure of DNA. Watson, Crick, and Wilkins did, but they used her image and they also used her data to come to those conclusions. Man, I can't finish these notes soon enough. I need to go brew myself a cup of coffee. Still got stuff to do today. All right, getting ready to hop ahead. Okay, so you don't necessarily need to have this in your notes, but you need to be able to recognize the parts of this. Hydrogen bonds, weak hydrogen bonds, connect the nitrogen bases. Okay, there's the sugar, 5 carbon, so there's going to be a carbon there, carbon there, carbon there, carbon there, carbon there. Sugar bonded to a phosphate, bonded to a sugar, bonded to a phosphate. This makes what is called the sugar phosphate backbone. So these ribbons are the sides of the ladder. Okay. And of course, nitrogen base, phosphate, sugar. These 3 things make up nucleotides. G always goes with C. A always goes with T. Hey guys, I'm recording a video. Please be quiet. Family just got home from shopping. And of course, with the new rules that are being set forth by Governor Newsom, it's going to be harder and harder to do shopping. Coming into the Christmas holiday season, do your stuff online and do it early because everybody's going to be doing their stuff online. So what did it lead to? Watson and Crick, Wilkins to a smaller degree, built a model that explained how DNA carried genetic information, and also kind of accidentally figured out that if you unzip the 2 sides of the ladder, they form templates, which is something that you use, and then enzymes bring in the new nitrogen bases and build a new molecule. So they also figured out that it's how DNA replication takes place, and they really kind of figured out the secret of life and how life propagates itself. Shape, double helix. So remember, Wilkins showed Photo 51 because he worked in the same place that Franklin did, showed Watson Photo 51. Watson did not have all of the experience with X-ray diffraction images, but Crick did. So he sketched what this looked like on his newspaper. He showed Crick, and Crick's like, I know what that is. That's a double helix. So that helped them figure out what their model needed to look like. Okay, base pairing. They found that H bonds, hydrogen bonds, form between A and T and between G and C. And they're weak hydrogen bonds. Excuse me. Moving ahead. DNA is made up of what? And what are these things made up of? If you can't remember, take a look back at number 16 and number 17 in your notes. That's why I number them, so it's easy for you to go look up these answers. looking them up, reading them helps you memorize. Yeah, so DNA is made up of nucleotides. Those are the monomers that make up the polymer DNA. And nucleotides have three parts: sugar, deoxyribose, phosphate group, nitrogen base. What are the names of the four nitrogen bases? And which ones bond with one another? If you can't remember, you might want to take a look at number 16 and number 20. 16 and 20. 18 and 20. My bad. 18 and 20. Okay, four bases are A, G, C, and T. A always goes with T. It's easy to remember that because AT is a word. Okay, and then, of course, if all you're left with is C and G, they have to bond with one another as well. A goes with T, C goes with G. Okay, Franklin found that the DNA molecules is what shape? If you can't remember, take a look at number 21. No, beg your pardon. Take a look at number 23. And then Watson and Crick built a model of DNA and showed that A and T nucleotides and G and C nucleotides are bonded together with what types of bonds? And you might want to take a look at number 24. Excuse me. Yeah, so it's called a double helix, which is a twisting three-dimensional ladder. And the nitrogen bases, or the nucleotides, are bonded together with weak hydrogen bonds. Okay, Avery and other scientists discovered that. Which one of these is correct? You got it. B. DNA stores and transmits genetic information from one generation to the next. The Hershey Chase. Oh my gosh. I thought I fixed these. Hello. Sorry. What just happened? Well, number one, I gave you the answer. Let's see. Come on, let me in there. Oh, that's what's the problem. Okay. Now, of course, I got to fix all this. Sorry. Cool. I got to click on this and make it bigger. And now, of course, I've given you the answer. All right. So, DNA has phosphorus, phosphate groups. Protein has sulfur. Of course, they found phosphorus, radioactive phosphorus, in the bacteria, meaning that the virus injected DNA and not protein. Sorry about all that chaos. DNA is a long molecule. What are the monomers of DNA called? Yep, they're called nucleotides. Chargaff's rules state that the number of guanine nucleotides must equal the number of G always bonds with, yep, cytosine. Last one, what are base pairing? What goes with what always? Yep, A and T, C and G. All right, ladies and gentlemen, that is it for the notes. Please make sure that you get these uploaded at the latest Sunday night at 9 o'clock. Make sure they're numbered. Make sure that they're question answer on the left, answer on the right. And we will see you back in the zoo.